Today, I'm going to talk about my group's work in studying, building a model of and studying the, oops, I'm just having all kinds of problems here. That was a video that should be going, there we go. Studying the JCVI-Syn3A using computational modeling. And one of the first questions that we wanted to think about with this was the idea that the nucleoid in Syn3A is expanded rather than what we call compacted as it is in many other bacteria. So nucleoid compaction in bacterial cells has been attributed to a number of things, cytoplasmic crowding, supercoiling effects, the action of nucleoid-associated proteins, among others. And in most bacteria, including E. coli, these mechanisms condense the nucleoid to a smaller volume within the cell, centralized, excluding most of the ribosomes to the surrounding cytoplasm. And in contrast, the nucleoid in 75 to 80% of mycoplasmas in which nucleoid localization has been studied, or at least reported, including the mycoplasma-derived Syn3A, these nucleoids span the entire cell with ribosomes distributed throughout. So that is interesting. In recent physically resolved whole cell models of mycoplasma-derived synthetic cells, introduced 10 base pair resolution models of the complete DNA in a simulated simple cytoplasm of cytosol and ribosomes, recapitulated separately, recapitulating DNA replication. And in tandem, Goodsell and Autin's work in 2024 also included RNA polymerase to visualize cell organization. And so these were really important sort of visualizations of spatial organization in Syn3A, something that is close to my heart to be thinking about physical distribution in these cells. And in both these studies, just because they were thinking about other things, they neglected other cytoplasmic proteins, electrostatic charge in these initial models, and nucleoid-associated HU proteins. Both studies also placed ribosomes into the simulation cell using an experimentally measured cell distribution. And in the work visualized here by Gilbert et al., the DNA was then self-assembled in the remaining volume. And because the space was available during the simulated self-assembly, a fully expanded nucleoid, makes sense. That sort of de facto had to take place. But if the model was then to incorporate other cytoplasmic proteins, we believe that these would tend to compact the nucleoid. So we thought, okay, including Crowder's and other basic effects like electrostatic charge would probably reveal more. So that was sort of our starting point. And then also in conversations with John, there was speculation emerging from recent studies, thank you, John, that suggests that Syn3A's lower concentration of NAPs underlie its expanded nucleoid. And we like to count. E. coli has more than 100,000 NAPs of various types, while Syn3A has about 30 of HU only. So that's about 50,000 NAPs per cubic micron and 43 base pair per NAP for E. coli, as compared to 300 NAPs per cubic micron and 18,100 base pairs per NAP for Syn3A. So I didn't want to just say the raw numbers, but we can see that when you account for size and size of the genome, that still there's a big disparity. So we decided to build a simple whole cell model of Syn3A to look at these effects. So today I'm going to talk about that recent work, examining several factors that appear to contribute to the expanded nucleoid of mycoplasma type cells, including charge and DNA bending HU proteins, which were neglected in the initial models. So to do so, we used a mesoscale resolution whole cell model of Syn3A that we built using Brownian dynamic simulations. And this is work led by my former postdoc, Dr. Jesse Roy, who's now an assistant professor at Villanova, and Dr. Vishal Sivasankar, who's an assistant research professor in my lab. So we built our Syn3A genome using John's genetic sequence, and we coarse-grained every 100 base pair into one bead, which we've since refined into 10 base pairs per bead. And we joined the beads together into a long polyelectrolyte bead spring chain in a Brownian dynamic simulation model. And that's where diffusion isn't something that we just input, but the action of the solvent causes physically explicitly represented individual particles to undergo a random walk. So the Brownian motion is first principles. Now, since the big question is whether the natural state of the nucleoid is expanded, we had to decide where to start. Do we put in the ribosomes first, DNA, or base pairs first? So as a starting point, we built a baseline model for Syn3A. Sorry, I'm getting over a cold, so my voice is kind of gravelly, and compared our molecular distribution to experimental results. And we used the ribosome data reported by Gilbert et al. And also captured in Goodcell's paper, which was previously used to inform their and other Syn3A models. So in these, the model Syn3A cell was built by placing ribosomes according to experiments, holding them fixed in place, and then letting the DNA assemble itself around it. So that was done previously. So we started by recapitulating that method using a swelling Monte Carlo algorithm to self-assemble the genome. So using this method, we generated initial condition shown here in these images through this recapitulation protocol, which gave the same almost uniform ribosome distribution and a cell-spanning nucleoid. And so there's a simulation snapshot shown below, and the plot shows the distribution of ribosomes in green and DNA in purple. And as with Gilbert et al. Model, the cell contains only DNA and ribosomes, nothing else, and entropic exclusion between them. So this approach recovers the experimental ribosome distribution for the initial state. But the prior model didn't include dynamics. That's not what they were interested in that time. And so we were curious, if we then letted Brownian dynamics to commence, not just a static model, we were concerned about whether or not this was a stable configuration. And so we let Brownian motion commence, which actually immediately redistributed all of the molecules. So this shows that simple entropic exclusion in Brownian motion leads to substantial migration and accumulation of ribosomes toward the cell membrane, and some nucleoid compaction, which actually disagrees with the experimental measurements. So obviously something is missing in such a simple model. So we decided to arrange the ribosomes in DNA. We said, well, maybe we should build it differently. And so we decided to arrange them without a priori knowledge of the experimentally measured ribosome configuration. And that resulting configuration is shown here, which is quite similar to the middle figure. So overall, in this very simple model with no charge and no Crowders, ribosomes diffuse naturally out of the nucleoid and accumulate near the cell membrane, compacting the nucleoid by at least 15% by volume. And this is inconsistent with the experimental observations of near homogeneous ribosome distribution in Syn3A, suggesting that such a simple model with only ribosomes in DNA and only hard sphere repulsion is too simple. So we already know that Syn3A has a lot of protein Crowders and an interesting charge distribution. So a baseline model should at least have those features, as well as effects of nucleoid-associated proteins. But one of the first things that we wanted to understand is how the inherent stiffness of the DNA affects things. So base pair stacking controlled by chemical bonds imparts DNA's intrinsic multi-scale structure and gives rise to its mesoscale material properties such as bending, stretching, and torsional stiffness. And as with non-biological polymers, we expect these intrinsic properties to influence overall compactness and size. The chromosome, which of course includes both DNA and bound proteins that can further alter local, global, and global stiffness, is described in our model using a dimensionless scalar coefficient, sigma S and sigma B, which set the chromosome's resistance to stretching and bending. And so here I'm showing our results for 3 values of bending stiffness, with the middle one, sigma B equal 10, being consistent with established literature for values for bare DNA, meaning no nucleoid-associated proteins. And we measured things like the radius of gyration and other metrics to show that this matched up with experimental results. And the top row is simulation snapshots, and the bottom row shows plots of probability of finding DNA and ribosome molecules from the cell center at the left of each plot to the cell wall, averaged over 100 realizations. So overall, no value of inherent DNA stiffness recovers the experimentally measured distribution of ribosomes. They all push ribosomes out to the cell membrane, and keeping in mind that that value of 10 is the one that's physiological. So we speculated that the nucleoid's inner pore size or mesh size distribution at least partly explains why most ribosomes cannot reside within the nucleoid. To characterize this structure, the porous structure in the nucleoid, we did a Voronoi-based analysis, which reveals pores of a range of sizes in the top row images, and the bottom row of simulation snapshots show progressively more heterogeneous structure as stiffness increases, which is interesting. This means that where the big pores are changes. So there's a concentration of many small pores and more DNA toward the center, and larger pores and less DNA near the nucleoid's edge, which is not surprising, along with a larger increasing DNA-free region near the cell membrane. So the light blue dashed line in each of these snapshots encloses about 95% of the DNA, illustrating sort of the global compactness in a visual way. We did measure the radius of gyration for each stiffness, and that's shown below each snapshot and confirms that lower stiffness of the DNA intends to expand the nucleoid region. The pore size distribution is shown on the top right in figure C for all three stiffnesses, and a single peak for each shows a dominant pore size, and the peak height and location and width all change appreciably for the less stiff nucleoids, with only about half of the pores able to exclude ribosome-sized biomolecules. And the pore size varies from the center to the cell membrane, and this is what's more interesting. And it changes as stiffness increases, as shown on the bottom right in panel D. And increasing stiffness drives down pore size near the center and drives pore size up near the edge of the nucleoid. So sort of the overall takeaway is that the presence and concentration of DNA segments alone does not exclude ribosomes, but rather the mesh structure permitted by the stiffness of bonds. In fact, the stiffest nucleoid's average pore size near the center is half the ribosome diameter, consistent with the exclusion of ribosomes that we've observed so far. And none of this is surprising, but we wanted to see that we could recapitulate what people have observed in other cells and experimentally. And we can use this same analysis, I won't go in deep into this, but we can use this to identify not just isolated pores, but more importantly, fully traversable pathways through the nucleoid that show that changed stiffness can cut off access of biomolecules above a certain size, which will affect gene expression. Okay, so now nucleoid-associated proteins, how do these help? So these can also promote structural changes in nucleoid by binding to DNA. And in contrast to other bacteria, Syn3A has only the one type, HU, which can make the nucleoid stiffer and produce local bends. Now, HU concentration in Syn3A has been noted in other mycoplasmas is notably lower than that of most bacteria, which might contribute to less nucleoid compactness or more expanded. So we decided to sort of test it and crank it up even unphysically so. We distributed local bending defects across the genome by changing the local equilibrium angle, and the number of defects was chosen to represent a range of concentrations from physiologically accurate to unphysical concentrations of HU, just to make the point. And the three simulation snapshots shown here show visually that increased HU concentration globally compacts the nucleoid, expelling ribosomes. So we again measured the spatial distribution of DNA and ribosomes in the three plots shown below, each of the simulation images, and the nucleoid pore size distribution, which gave a similar effect as increased stiffness, confirming HU's mechanistic action mechanically. And notably, the physiological concentration of HU proteins, NHU is 50, doesn't do much to compact the nucleoid. So overall, this supports the recent hypothesis that the paucity of HU proteins in Syn3A contributes to its nucleoid self-spanning structure. However, HU proteins are a small portion of all the proteins in Syn3A. So up to this point, our model, similar to the model from Gilbert et al., did not include cytoplasmic proteins. This produced the leading order effect of lower cell packing fraction. But here we examined whether the presence of a secondary size population, so polydispersity, induces additional effects. It's an entropic effect, and it's well known that crowding from cytoplasmic proteins drives nucleoid compaction. So we expected more packing to further compact the nucleoid and worsen our agreement with experimental prediction of the cell-spanning nucleoid. And the top row is the original model that we've been talking about, and the bottom row is at the same packing fraction, but now with the physiologically accurate relative abundance of cytoplasmic proteins. So this more crowded physiological composition of proteins, ribosomes, and DNA actually markedly compacts the nucleoid and expels ribosomes out of the center of the cell toward the membrane. And so the DNA-depleted annulus near the cell membrane is also widened. So overall, these results confirm that the physiological abundance of proteins, which is often neglected in simulations, is a big deal. And if you include it, it worsens agreement with experiments. And so the next thing to bring in is charge. So to probe the effect of electrostatics, we endowed each biomolecule with surface distributed electrostatic charge. And all DNA and ribosomes in the model have uniformly distributed negative surface charge. That was just sort of the initial way we wanted to do the model. using physiologically accurate values taken from literature. And for the protein surface charge, we determined the solvent accessible residues of the folded protein structure and evaluated its dissociation constant to determine the pH dependent net charge. So notably, up to 75% of Syn3A's proteins are positively charged, with 25 to 30% negatively charged, and most of them weakly. This is almost opposite to E. coli, whose proteome is about 40% positively charged and 60% negatively charged. So I'm going to set that off to the side for a moment. On the left, I'm showing a simulation snapshot split into two hemispheres, with some molecules in the right half made invisible to reveal the nucleoid. And there you can see aggregates and condensates have formed due to the interactions between positive and negatively charged proteins and ribosomes, and proteins and proteins. And there are several exploded views shown around the main cell to illustrate the range of composition and the size of these condensates. So these sizes and compositions are analyzed in the plot at the top right alongside the uncharged system, and unsurprisingly, electrostatic interactions drive formation of small, medium, and large condensates. Some are proteins only, and others are proteins bound to ribosomes, which is an effect observed in other bacteria, in fact, in the study that we did recently on E. coli. So what's interesting is now negatively charged ribosomes are present deep within the negatively charged nucleoid. An analysis of these ribosomes revealed that they are part of condensates. They are coated with positively charged proteins. So what that means is that proteins are basically cloaking ribosomes and allowing them to go into the nucleoid. The bottom right plot shows a substantial population of these ribosomes inside the nucleoid, agreeing with the experimental evaluation, finally getting some agreement with those experimental measurements of Syn3A and mycoplasmas. So sort of takeaway is that evidently charge shielding of the ribosome can drive their cell-wide distribution in Syn3A and maybe other mycoplasmas. And these results are consistent now with our study also of E. coli. So that's the key new result, is that the electrostatic charges on proteins, DNA, and ribosomes causes the nucleoid to expand throughout the cell. And one key change is a reduction of DNA near the center. A shift of several thousand beads from the center redistributes to the annulus near the membrane, which, because it has more volume, doesn't look so much big change graphically. Okay, so overall findings and takeaways. There's an interplay between DNA chain stiffness, HU-induced bends, cytoplasmic crowding, and electrostatics that regulates the distribution of DNA, ribosomes, and proteins in our Syn3A model. The nucleoid stiffness, HU concentration, protein crowding all enhance nucleoid compaction, but electrostatic interactions are the only mechanism that we studied here that counteracts compaction. And specifically, the atypical charge distribution in Syn3A, where the majority of proteins is positively charged, allows more ribosomes to interpenetrate the nucleoid by shielding their negative charge and thus expanding it. And I want to conclude by acknowledging our funding sources and high-performance computing sources and collaborators.
Okay, so with Syn3A, we have a unique situation where we have fundamentally eliminated the nucleoid proteins, except for a tiny number which may have some requirement, for instance, maybe at the origin of replication or something.
Okay.
Is from perspective of understanding cell biology, should we be more interested in this aberrant situation, or would it, can you justify the value in studying that relative to what is true in presumably all other cells in our world, that the DNA is bound up in nucleoid?
So let me read back your question, make sure I understand it. So you recently did some studies where you pretty much got rid of HU, is that right? Or you?
Yeah. The minimal cell has 300-fold less.
Oh, I see what you're asking. So was this idea HU that you eliminated everything except this small amount of HU? Is it realistic what you did is it realistic to have made the minimal cell have just this small number of HU proteins? Is that the question?
Well, I'm thinking more about from a perspective of your modeling. So yeah. Should we be focused on this aberration or should be, I mean, it seems like you need to be looking at how does this make cells different than what we see in modern cells?
Yeah, I think what's interesting is that in modern cells, we have the examples of the mycoplasmas, and they also similarly have a fairly low abundance of nucleoid-associated proteins. And they also, except capricolum has been reported to have a more condensed nucleoid, although I would like to see more than one data point on that. Whereas 4 other mycoplasma studies that we were able to read experimental reports on show the expanded nucleoid. And so this idea of, I would look at it as this is maybe sort of a strategy. And by the way, at the same time, there's not, to my knowledge, evidence of polysome formation. So I think that this is, I wouldn't say that this is something else that's an incorrect focus, because it's present in other mycoplasmas. To me, it seems like a strategy, a survival strategy.